everyone's here, huh? So, let's go ahead and get started. Before we get into today's chapter, one announcement. So, I have an unavoidable dinner that I have to do Monday night, okay, after spring break. I know you guys don't have class on Monday, but since I'm canceling class on Monday, I'm going to also cancel the Tuesday class for you guys, just so you can. That way we're on the same page. So what that means for us is that class will be canceled, and we will just push everything down one class. And then where we'll make up our lost time is on April 4th. So April 4th, Disorders and Immunity, Chapter 16 will be cut out, and the Parasites Lab will be cut out. Okay, both of those are expendable for me just in case something like this happens. I design it that way.
So which day is it canceled?
So the day that's canceled will be the 21st of March, okay, which is the day after spring break, okay, or class after, I should say. So that means you guys get a little extra long spring break period. And then we'll just continue on. So we'll do chapter 12 then on Thursday. And then we'll do chapter 13 and 14 and 15. Chapter 16, though, will be cut out. Okay? And that's where we'll make up our lost time. Any questions on that? If you know anyone that's not here, please let them know. Okay, so they don't just show up.
What about that?
Yeah, no. Well, you'd be surprised. If they come, they show up, and no one's here, then you get people that want to do that.
You know, like I was going to do at the beginning of this.
She thought I had a mental break.
break.
And I go, what are you talking about? We don't start before tonight. So I'm seeing you.
Yeah, this
And I'd be the one sitting there going,
this is the only campus that does it. For some reason. I don't know, but I like it. It's longer time off for me.
I mean, it took until, took me forever to get it through my head that.
So we're going to do 12, 13, are we in 14?
Yep, 12, 13, 14, 15.
Oh,
Okay, 16 is the only one we're cutting out.
Okay.
Everything just gets pushed down a day until we reach the 16th day, and then everything goes back to normal. Okay? Alright guys, so chapter 10 deals with genetic engineering and recombinant DNA technology. Okay? Essentially, this is a chapter that talks a lot about techniques that we do in the laboratory to manipulate DNA. Okay? Essentially, what we'll talk about is how we can manipulate DNA and then put it into organisms so that they make whatever that DNA codes for. Okay, that we manipulate. This is a very important technique done in labs, not only for basic science, like what I do to understand how organisms work, but also to produce proteins for clinical therapies. Okay? So it's an important concept, but with that said, it's a pretty dense concept. Okay? So I don't really expect you to take in a lot of this and be able to regurgitate it in an exam. However, there are a couple key points which we'll point out that are important, okay, that you know. But a lot of this is going to be for your information, more so than actually testing you on. Also, because it is very technique-driven, this is stuff that you would see on the second practical, okay? Not so much in the next lectures. So, genetic engineering is essentially artificial manipulation of genetic material, okay? Primarily DNA, but it can be RNA as well. Now, this is a little complicated because DNA is very small and hard to see, okay? So we need to use special tools, primarily enzymes, to manipulate and chew up this DNA in the way that we see fit. And we do this to answer both basic science questions and applied science questions. Basic science questions are what I do, okay? This is what we call basic research. It's not basic because it's simple, it's basic because it's fundamental, okay? And what we're doing is we are, manipulating the genetic material in an organism and then seeing what that does to the organism, okay? And this can then answer questions about that organism, which we can utilize later. Applied sciences is where you take some of these concepts we learned in basic science and then, scale it up and then use it like in a clinical setting, for example, okay? So this approach can be used in both of those applications. Always starts with basic and then it works its way into the applied science field. Some things that we can do with genetic engineering would be like characterizing organisms, if you're a basic scientist, using it to identify an organism, if you're a clinical microbiologist, or you can use it to actually modify organisms, or what we call genetically modified organisms, and this then makes the organism more useful. Okay? It may produce a protein that is, very nutrient rich and makes it more, palatable for humans or something along those lines. So to do this, what we're going to do is talk about 6 different kind of topics. We'll start off with tools and techniques, and then some of the methods, and then how we put all these methods together to eventually end up with genetically modified organisms or treatments or even genetic analysis. So first off, we have to talk about the basic molecule that we're going to manipulate, and as I said before, this is almost always DNA. So we start off with DNA, and then we have to figure out creative ways of changing it or modifying it.
It's going to be the 8th edition of
And as I said, DNA is very small, so it's not like we can just pull it out and cut it with scissors and tape it together. Instead, we have to use enzymes to do the work for us. Okay? These enzymes are known as restriction endonucleases and ligases. Restriction endonucleases cut DNA like scissors, and ligases glue them together like glue. Now, DNA, as we talked about before, is a double-stranded molecule. It's helical in nature. And those 2 strands are held together by weak hydrogen bonds. And in Chapter 9, we talked about how there are enzymes like helicase which can split these 2 strands apart. And this is important because DNA isn't very useful when it's double-stranded. It has to be split apart to be useful, especially by organisms. Now, in a cell, we use enzymes to split the strands apart. But in a test tube, instead, we use heat. Okay? And this is a process called heat denaturization. This is where if we heat the DNA up to about 95 degrees Celsius, or right below boiling, the 2 strands will fall apart. Okay? And that's because the hydrogen bonds are too weak and they just separate. What's really cool about this is that if we take that denatured DNA and then we cool it back down to room temperature, those 2 strands will zip right back together. Okay? This is a process known as annealing. So by doing nothing but add heat or take away heat, we can pull the DNA apart or put it back together, depending on what we're trying to accomplish. Okay? So as I said, we do most of the work with enzymes, and some of the most important enzymes are going to be restriction endonucleases. These are enzymes that literally cut the DNA, kind of like a pair of scissors. And these restriction endonucleases cut the DNA at very specific sequences, okay? Anywhere from 4 to 10 nucleotides. And so whenever they see their particular sequence, they cut, okay? And this is useful for us because we can look at a piece of DNA that we want, and we can find one of these cut places in the beginning, one of these cut places at the end, add in those enzymes, cuts it up, and then we can pull out the fragment of interest. Okay, so just like a pair of scissors. Where did these come from? So restriction endonucleases actually come from bacteria. These are thought to be rudimentary immune systems for the bacteria. So the sites that these cut at, these sequences right here, are not found anywhere in the bacterial genome. Okay? So they don't cut the bacterial's genome. But let's say a virus comes in and injects DNA. If that virus has one of these sites, then these guys will chew up the viral DNA, and then the virus doesn't infect the bacteria. Okay? So we can isolate these from bacteria, and we can utilize them in a laboratory setting. There are hundreds of these available, and they're usually named after the organism they came from. Okay? So BAMH1, which is a very common one, is named after this bacillus organism. And so whenever this enzyme sees this sequence, it cuts the DNA. Now, interestingly enough, with this enzyme in particular, it cuts and creates what we call a sticky end. All that means is it doesn't cut straight down, it cuts asymmetrically. And what you're left with are little pieces of single-stranded DNA that kind of hang off the side. Okay? We call these sticky because they can stick to other pieces of DNA. And so we can use that to kind of stick these guys together. EcoR1 from E. coli, this guy also has an asymmetric cut, or a sticky end. But not all of them do this. Some of them cut as what we call blunt cutters, and that means they go straight down. There's no sticky single-stranded ends. EcoRV is a prime example, and you can see it goes straight down the middle. So we use these to cut the DNA and then excise or remove that DNA of interest. Okay? And if we do this in such a way, creating particular sticky ends, those little pieces of single-stranded DNA, we can then take that and stick it to another piece of DNA that we're interested in creating. Okay? If we do this over and over and over again, we'll create an artificial DNA sequence. Now, some other enzymes that we use, so ligase, as we mentioned before in chapter 9, ligases are used to fill in any gaps between DNA. Okay? So after we get our DNA stuck together, we'll add in a ligase, and this will fill in all the gaps so that it's strong, it doesn't fall apart. Another enzyme that gets used when we're dealing with eukaryotic organisms is an enzyme called reverse transcriptase. This is an enzyme that's naturally found in HIV, okay, or what we call retroviruses. And this is an interesting enzyme that converts RNA into DNA. Okay? Now the reason why we have to use this in eukaryotes is because let's say we want to make a gene or make a protein from a eukaryotic gene. Okay? If you didn't really think about it that hard, you would say, okay, I'll go into the organism's genome, right, pull out its DNA, and then we'll cut out the gene and just stick it into another organism. And in theory, that makes sense. But there's one problem that develops if you do it that way. And that is, in the DNA side of things for eukaryotes, there's all that garbage DNA that we talked about. Okay? These are called introns. So if I were to just cut out my gene from the DNA side and put it into an organism, all that garbage would come with it, and it wouldn't work. But remember that the cell gets rid of that garbage once it gets turned into RNA, and then it splices that out once it leaves and goes into the nucleus. So how do we get around this? Well, what we could do instead is isolate the RNA from the organism and convert that into DNA. By doing it this way, we're now getting rid of all of that garbage intronic DNA because the cell cut it out for us. Okay? So this is what has to happen when you're dealing with eukaryotic cells. An example of where this has worked very well for humans is human insulin. Okay? Human insulin, there's a gene for it. If you pull out the RNA for a human insulin, you can turn that into cDNA, or just copy DNA of the RNA. Then make that double-stranded, stick it into a vector, which we'll talk about in a little bit, and then put that into yeast. You then grow the yeast up, they make the human insulin, you pop them open, pull out the human insulin, and then you inject it into your leg. Okay? So you can do this to make practical proteins that are useful for humans, especially in clinical disease. Before this, insulin primarily came from pigs. Okay? It's close enough, but not really the same thing. So it didn't work as well. Now that we can make human insulin by using this approach, it's much cleaner and much better for the patient. Now, we've talked about using enzymes to cut DNA into pieces, but after we do this, we want to make sure we have the right piece. Okay? So we need a way of visualizing the DNA. As I mentioned before, DNA is very small, so you can't just see it even with an electron microscope. So we had to get a little creative. And the primary way that we do this is by using something called gel electrophoresis. This is where we take our DNA, which has been cut up into little pieces, thanks to our restriction enzymes, and we add this to a gel. Okay? And this gel is essentially agarose, which is pretty much the same stuff that we use in lab in our petri dishes. Okay? And this agarose, when it solidifies, it creates a sieve, okay, or a bunch of little holes in it. So when we add our DNA to this agarose, and then we pass an electric current through it, this electric current will push the DNA through that sieve. And the big pieces of DNA get stuck up at the top, and the small pieces of DNA get stuck down at the bottom. And in this gel, we add in a chemical called ethidium bromide. This binds to DNA and makes it glow under UV light. So what we can do here is we can add our DNA, run it through the gel, stick it on a UV table, shines UV light through it, and we can look and see if we see any fluorescent bands. Those fluorescent bands are our DNA pieces. And what's really cool about doing it in this way is that not only can we see our DNA bands, or pieces, we can actually determine how big they are based on where they are in the gel. If they're up at the top, they're really big. If they're down at the bottom, they're really small. Why is that useful? Well, let's say I cut out a gene and I know it's exactly 1,000 nucleotides. Well, if I run it on a gel and it lands right where 1,000 nucleotides is, that's probably my piece, right? But if it lands somewhere way up here or somewhere way down here, then maybe it's not. It could be something else, okay? The biggest problem with this approach is that you need a lot of DNA, okay? You can't just use a little bit. You need a lot to be able to visualize it. So now we have a way of separating out our DNA based on size, okay? And actually visualizing it. But what if we want to know exactly what piece of DNA we're dealing with, okay? One of the easiest ways to figure this out is to use a concept called hybridization. And this is where DNA will stick to itself as long as the bases complement each other. So remember before we talked about how C's always stick to G's and T's always stick to A's? So let's say I'm trying to find the human insulin gene again, okay? And if I look at the human insulin gene, I know that it has this sequence. I'm just making this up. I can, using chemistry and machines, okay, develop a small little piece of DNA that binds to this sequence, okay? And it's all based off of C sticking to G's and T sticking to A's. So if I were to design one of these, let's get a different color. I would design it so that it looked like this. They stick to each other. That's great, right? They stick, that's wonderful. But how do I actually see this? Well, on this little piece of DNA, which we call a probe, I would also add something I can easily detect to it. Traditionally, this is a radioisotope, which is radioactive, it emits particles, and you can detect it with a Geiger counter. So if I run a Geiger counter over my band and it goes din 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 din, it's done. That's it. I run it over and I don't get the din 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 din, it's not. The more modern technique, since radioactivity is kind of dangerous, is to use fluorescence. This is a molecule that is boring by itself, but if you shine UV light on it, it emits light. So we can then easily see it. Using this approach, we can tell whether or not our band is present. Okay? If this guy sticks and we see the fluorescence, it's present. If this doesn't stick, we shine UV light on it, it won't glow because this fluorescent molecule isn't there, and that tells us that's some other weird piece of DNA and we have to go back and figure out what went wrong. Now, the old school way of doing this was using something called a southern blot, and this is where we run our DNA using electrophoresis, like we talked about, so everyone's separated by large to small, and then we would mix it with our probes, and then we look and see where those probes bound. Wherever they bound, that tells us that's where our DNA is, and then we cut that out and use it. Okay? We can also use this for diagnostic approaches. So one kind of cool way of doing this is developing these probes so that they only bind to a specific organism's DNA. Okay? Every organism has unique DNA. So you can design these probes so that they only stick to a particular organism. Let's say for this example, Campylobacter. Campylobacter jejuni causes diarrhea. Okay? So if you want to test for Campylobacter jejuni, what you could do is you could take a stool sample and you could streak it out on an auger. Now you're going to get a lot of colonies, right? Because there's a lot of bacteria in our gut. So how do we determine which one's Campylobacter? Well, what we would do is we'd take a piece of filter paper, we'd stick it on top of these organisms so that some of them stick to the paper. Okay? We then lyse the cells, pop them open, and open up their DNA using sodium hydroxide, or a basic solution. We then take this and we add our probes. Okay? In this case, it's going to be a probe that only binds to Campylobacter's DNA. We then wash off anything that didn't bind, and then we detect whether or not that probe is there. In this case, out of all of those colonies we had with, 2 of them show that the probe is there. We then take that and we match it up to our original plate, and we pick those 2 colonies. Those are the guys that cause the disease. Okay? So you can use this for diagnostic approaches as well. If you want to actually look at whether or not an organism has a particular piece of DNA in it, within the organism itself, without lysing it and popping it open and doing all this crazy stuff, you can do something called fluorescent in situ hybridization, also known as FISH. Okay? What this does is it allows you to add your probe to cells that are still intact. So the reason this is cool is then you can look under the microscope and actually determine their size, their shape, are they gram-positive, are they gram-negative. You can also potentially take it from infected tissue. So I could take a biopsy of the infection, and then I can add in my probes, and wherever it glows, that tells me there's my bacteria. Okay, so it tells me exactly where they are within the tissue. All right, so, yeah.
Quick question. Would this be used like to diagnose, like if you have an upper GI, would that be something, like when you drink the, it's like a radioactive treatment, is that what it's doing? How it, you can see little ulcers?
So what that typically does is that's a contrast dye, and what it's primarily looking for is high areas of blood flow. Okay? And so if you have an ulcer, you're going to have more blood flow in the area because it's a problem, right? You got inflammation and things like that. But this does get used for diagnostic purposes, but it usually requires you to have a biopsy or a sample that you do outside of the body. Okay? And this FISH technique is actually really simple to do. So you can do this with, a cheap microscope in sub-Saharan Africa. So this is becoming way more common nowadays because it's a very cheap and easy approach that can be done. Examples of where it gets done. So syphilis, right? Syphilis is a small organism. You can't see with the normal light microscope. But if you do FISH, you can see it real easily because it glows. It's easier to detect. Okay, so that is using hybridization probes. Now, hybridization is great, but there's one problem with hybridization, is you have to know the sequence of your organism, right? I have to know that this is only found in Campylobacter. Otherwise, I can't do it. So how do we determine what the sequence of our organism is? This is a process known as DNA sequencing. Okay? And DNA sequencing literally does exactly what its name describes. It tells you the sequence of the nucleotides. Now this is a very useful tool. Not only does it tell us this so we can design probes, but it tells us a lot about the organism. Okay? If we get the entire sequence, let's say from a species of E. coli, we can take that sequence, put it into a database, and that database will compare it to all other organisms that we know of or have sequenced. And then at the end of the day, it will spit out a percent identity, which means how similar is that DNA compared to the known DNAs. And let's say that the unknown species of E. coli comes out to be 99.9% identical to a pathogenic strain of E. coli. Well, you can say, okay, that's very similar, which means it might be pathogenic. It might cause a problem. Not only can we compare it to known samples, we can actually look at the DNA and we can learn more about the organism. We can look at it and say, oh, look, this guy has a gene in it that makes it resistant to, chloramphenicol, which is an antibiotic. So now we know, don't treat with that antibiotic. Okay? So sequencing is a very important technique. It used to be really expensive and difficult to do, but it's becoming much, 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 much cheaper nowadays. And so I can guarantee you within the next 10, 15 years, you will be seeing this popping up everywhere in the clinical world. Okay? Now, the traditional way to do DNA sequencing is something called Sanger sequencing. And Sanger sequencing is a relatively complex process, so we won't go through the details, but it uses fluorescence to determine the A's, the T's, the C's, and the G's, okay, in the sequence. And so by the end of the day, this is what you would get. Okay? And what you see are all these little peaks. Each one of these peaks represents a single nucleotide. And based on the color of that peak, it tells you what the nucleotide is. Okay? Now, Sanger sequencing is nice, but you can only do about 300 to 800 base pairs at a time. which sounds kind of like a lot, but when you think about a human that has 4 billion, that would take a very long time to get all of them. In fact, it took us 20 years to do it using Sanger sequencing. So Sanger sequencing is great for little stuff, but if you want the big stuff, you've got to use something called next generation sequencing. And this is where you can do a whole human genome in about a day compared to 20 years. Okay? It's much more efficient. It's also a hell of a lot more cost effective. So now we can figure out our sequence. Another really important technique once we figure out our sequence is being able to make copies of it. Okay? So if you take one cell and you have one piece of DNA, that's great. But as we said before, oftentimes we need a lot of pieces of DNA to do stuff with it, right? Even to just look at it on a gel. So one of the easiest ways to make copies of it is to use something called polymerase chain reaction, or PCR. This is essentially a Xerox machine for DNA. It makes specific copies of DNA, and it does it a million times. Okay? So if you start off with 1, you'll get 1 million at the end of the day. It uses enzymes and little pieces of DNA called primers. Spoiler alert, primers are the same thing as probes. Okay? They're just small little pieces of DNA that complement the region of interest. The only difference between a probe and a primer is that in PCR you need 2 of them, not just 1. So let's briefly talk about how PCR works. So this is one of the techniques that I do expect you to know for the exam. Okay, so make sure you pay attention. So the first thing we start off with is our DNA. Okay? So our DNA, we're isolating this from an organism, so it is going to be double-stranded. Now, the very first step in processing this or copying this with PCR is to denature it. Okay? And that's where we heat it up to 95 degrees Celsius. And remember earlier in the chapter I said when you heat DNA to 95 degrees Celsius, it splits into 2 single strands. Okay? So after this process, we end up with 2 single-stranded pieces of DNA. Like so. So that's step 1. The next step, step 2, is something called annealing. And this is where our primers can bind to the single-stranded DNA. Okay? So we've got our single-stranded piece of DNA. The primers that I put in are small little probes, like we talked about before, that bind in 2 places on the DNA. One at the beginning of the sequence we want to copy, and one at the end of the sequence that we want to copy, but on the bottom or lagging strand. Okay? Everything in between these 2 is what will get copied. Everything else will be ignored for the most part. Okay? Now, not only can we use this to amplify up sequences for genetic engineering, but because we can design these primers in such a way that they are very specific, we can actually use this for identification of organisms as well. If I design my primers so they only bind to Campylobacter, then only when Campylobacter DNA is present will this process work. Campylobacter isn't present, it doesn't work, and I get nothing at the end of the day. So you can use that as a clinical approach for diagnosis. And nowadays, this is a very common technique done for diagnosing in the clinical setting. Okay? And that's why I think it's important for you guys to understand it. So you may ask, how do we allow this annealing to happen? Well, this all happens based on temperature. What we do is we go from 95 degrees to 50 to 60 degrees Celsius, depending on our primer. And this drop in temperature allows for these little guys to stick. Now, the last step is what we call extension. And this is where we have our single-stranded DNA, or two single-stranded pieces of DNA. We've got our primers stuck to them, okay? And next comes an enzyme known as Taq polymerase. What this enzyme does is it binds wherever the primers are, and it adds in nucleotides, creating two double-stranded pieces of DNA. And just with the third part?
Yeah.
So the third part is where we have an enzyme called Taq, T-A-Q, polymerase, which binds to wherever the primer is, and then goes down the DNA, adding nucleotides, making it double-stranded. Okay? How do we control Taq? Well, Taq is from an organism that likes heat. More particularly, it likes 72 degrees. So only at 72 degrees will Taq work. So all we got to do is go from 50 to 60 up to 72, and it starts working. Yep. So we have special machines called PCR cyclers that can go from these temperatures very quickly, okay, to allow for this to occur. Now, at the end of the day, after these three steps, we go from one double-stranded piece, to two double-stranded molecules. But I'm pretty sure earlier I told you this makes a lot of DNA. How do we end up making a lot of DNA from this? We repeat this 35 to 40 times. So that for the next one, we now have two molecules we start off with instead of one. And then each one of those get copied, and so we end up with 4. Then the next one, the 4 become 8, 8, 16, 32, 64, 128, yada, 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 yada. Okay? So it grows exponentially, which means you get a lot of DNA at the end of the day. Now, as I said, this is important for genetic engineering so that we can get our pieces of DNA that we're interested in. But it's also really important for the clinical world, because you can design this in such a way that it only works when a specific organism is present. And the main benefits of this are huge. First of all, you don't need much organism to do this test. Why? Because you only need a couple pieces of DNA. So you can literally go straight from a swab in someone's throat to a PCR test and get a result in a couple hours. Also, you don't have to keep the organism alive, because you don't have to culture it. So it makes it easier for nurses to take the samples and collect them without having to make them, grow and auger and things like that. So it makes it very, very easy to get a good clinical diagnosis within hours. So this is what it looks like from the book with a little cartoon diagram. So again, denaturization, you get your single-stranded. You then have your annealing of your primers, which bind at two sites, at the beginning and at the end, the region you want to copy. And then you have extension, which is where you have your polymerase binding, wherever the primer is, and then adding on nucleotides, creating a new double strand. And if you do this over and over and over again, it grows exponentially until you have millions of copies of DNA. Okay, so those are the basic techniques. Now let's talk about how we can take all of these to eventually make something useful in microorganisms. So what the name of the game is for recombinant DNA technology, which is essentially making artificial DNA in the lab and then putting it back into an organism, is to select a gene of interest, excise that gene, isolate it so it's by itself, and then insert it into something called a vector. A vector is essentially an artificial plasmid. So remember we talked about plasmids in chapter 4? Those are circular little pieces of DNA that bacteria can shovel back and forth between each other. Well, a vector is the same thing. It's a circular piece of DNA, but it's artificial. We made it in the lab. Okay? What this does is it stabilizes our gene and makes it easier for the cell to use it. Once it's in the vector, we then stick this into a cloning host, which can be a bacteria or a yeast, or really any organism if we try hard enough. The organism then picks up that vector. It can then transcribe and translate it, like we talked about in Chapter 9, and then eventually make the protein for whatever that gene codes for. So, the first thing we have to do is obtain the target gene. And we're going to obtain this from whatever organism we want the protein to come from, right? So if it's for humans, it's going to be from a human cell. If it's for cows, it's going to be from a cow cell. We then isolate that gene, okay? And we slap that into our vector. Okay? And again, that's just an artificial plasmid. We then take that vector and put it into the organism. The organism transcribes, translates it, and makes said protein. And we can isolate that protein and utilize that to treat patients, right? So an example would be insulin or estrogen or human growth hormone. We can also take that DNA and put it into the germline of an organism, okay, or a little baby organism. And so that every time that that organism grows and divides into a higher order organism like a plant, every single one of those cells in that guy will have that recombinant DNA and subsequent protein production. This gets done a lot with plants. Okay? We call these transgenic plants, and we can literally put in any protein we want into these plants. Some examples of proteins we would put in, so pesticide resistance or resistance to pests, so it's literally a pesticide. Herbicide resistance, or the ability to survive in the presence of things like Roundup, which kill weeds but not the plant of interest. But we can also make the plants more nutritional. So we can add in vitamins, vitamin B12, so that it's healthier and easier for people that rely only on that plant to get all the nutrients required. And then lastly, we can use this technology to understand more about the organism. And this is the basic science side of it. This is where we, add things or delete things and then see what happens to the organism. And then we can infer the importance of those from that approach. So first off, where do we get our genes from? Okay. So as we talked about before, if you're dealing with prokaryotes, you get the genes from the DNA, okay, from the genome. The old school way was to cut it out and then do a southern blot, but again, this is old school. We don't really do that anymore. Instead, most of the time, we amplify it with PCR. So if I want a prokaryotic gene, I pull out their DNA, and then I design primers that bind to either end of the gene and make copies of it. Okay? It's as simple as that. For eukaryotes, it's a little bit more difficult. Again, it's because of all that weird splicing that has to happen. So we have to isolate the RNA, not the DNA, and then turn that into DNA using reverse transcriptase, and then do the PCR for the gene. Okay? So for eukaryotes, there's one additional step to get rid of all that intronic or garbage DNA. Once we get our gene, we then put it into a vector. And again, vectors, as we mentioned before, are just artificial plasmids. And they contain a couple different things. Okay? A basic vector has to have a couple of things. First off, you need to have something called a promoter. And a promoter is a region of DNA that allows for transcription. Okay? So without the promoter, the gene will not get turned into RNA. So you need to have the promoter so that it becomes RNA in the cell. Not only are there promoters, you will also have an origin of replication. We talked about what origin of replications are in Chapter 9. These are regions of DNA that allow the cell to make copies of it. Okay? The reason why this is beneficial is, let's say you only get 10 of these into the cell. Well, once the cell takes it up, it sees the origin and then makes thousands of copies of it. So now instead of having 10, you've got 100,000, which means you'll get more stuff at the end of the day. And the other thing that has to be found within a plasmid is a resistance gene. Okay? This resistance gene is a gene that codes for resistance to a particular antibiotic. In this example, this is a resistance gene to ampicillin, an antibiotic. And you may say, why do we include this? Well, getting this plasmid, this vector, into a bacteria is very difficult. It's very inefficient. So maybe only 1 out of 100,000 bacteria actually get this in them. So we need a way to select only for the organisms that get this DNA in them. So how do we do that? We have this resistance gene. So when the organism does pick up the DNA, if it does, it's 1 out of 100,000 that do, it now is resistant to ampicillin. It can grow in the presence of it. So if we take these guys that we put the plasmid in and put them on ampicillin, only the ones that have this in them will grow. Everyone else dies. So we select for the organisms that have this DNA in it based on this resistance gene. So how exactly do we put in all of our gene into the plasmid? Well, we do it using restriction endonucleases. They are those enzymes that cut DNA at specific sequences. So in this example, we have our gene of interest, and we have our plasmid that we want to put it in. What we do is we find sites or enzymes that can cut on either end of the gene, as well as either side of the plasmid. And we cut both of these with the exact same enzyme. Why do we do that? Well, once these are cut, you end up with those sticky ends or those single-stranded bits on either end that are complementary to each other. So you cut with the same enzymes, you then mix these two pieces that are cut up together, and they just magically stick together, thanks to the complementarity of the DNA. We then add in our ligase, which just fills in the gaps and makes sure it's really strong, and then we are left with our vector, with our DNA inside of it. We then take that and do something called a transformation, which is where we stick it into an organism. The organism then transcribes and translates that into protein, which we then can isolate and utilize later. This whole process from start to finish takes about 2 weeks. Okay? But you got to let things grow in between and all that stuff. Sounds like kind of a long time, but if you think about it, we are taking just a piece of random DNA, manipulating it like crazy, and then putting it into an organism so it makes something that it's never made before. Okay, so it's a relatively, pretty important thing to do in only 2 weeks. And again, we select for the organisms that have the DNA in them based on that resistance gene. Okay, so in the plasmid, they have a gene that makes them resistant to a particular antibiotic. So if the organism has the plasmid in it, it survives when that antibiotic is there. If the organism does not have that gene in it, and you plate it, it will die. Because the antibiotic kills it, and it has no way of getting around it. Okay? So this is important because, again, it's a very inefficient process. So from this step down here to here, we are actually going to do this in lab. Okay? We are going to take E. coli, we're going to poke little holes in it so it's easier for the DNA to get in. And then we're going to take an already made plasmid and put it inside of our E. coli. We then will plate it on a plate that has ampicillin, and what you'll see is only a couple colonies grow. Those are the colonies that took up the plasmid, or the DNA. How do we know that? Well, not only did they survive the ampicillin, but in this plasmid we have a gene called GFP, which stands for green fluorescent protein. So not only will they have grown on the ampicillin, when we shine UV light on them, they will glow green. Okay? So how does this get used in the real world? As I mentioned, you can make a lot of different proteins from this. Okay? And a lot of these can be human proteins, which can be useful in treating genetic diseases. So you can make a lot of immune treatments like interferons and interleukins, okay? Chemicals that are used for signaling in immune cells. You can make things like erythropoietin, or EPO, which is used to make or boost red blood cells. This can be utilized as a drug in the athletic world, or it can be utilized for people that have anemia, okay, or low red blood cells. What?
Just found out that iron's like an 8.
Oh, really? Get you some erythropoietin. You can make things like human growth hormone, which help people who lack this for whatever genetic reason. Makes them dwarfs when they don't have the ability to produce this. So if you add human growth hormone, they can grow at a normal rate. You can also make things like estrogens, for example, which can be utilized to treat when you're going through menopause and things like that. Before this, interestingly enough, estrogen came from horse urine. So this is a much more efficient way of getting estrogen. Not only do you not need to collect horse urine, it's actually human in origin. It's not from horses. Now, not only can we use this to make protein, okay, we can also take this DNA that we create in the lab and stick it into an organism permanently, okay? And if we do this in the germ cell, which is the precursor cell to a big organism, every single cell that gets produced from that will also have the DNA inside of it. This is what we call a genetically modified organism, okay? So if we put it in a baby cell, every cell that comes from that baby cell also has that protein. Where is this used for prokaryotes? Well, we can take certain species of Pseudomonas organisms. We can put in proteins that, let's say, are resistant to freezing, okay? And now we have an organism that doesn't freeze. And you may say, okay, who cares? Well, if you take that organism and then spray it on plants right before a frost, it stops the frost from freezing the plant and then subsequently killing it. And it's sort of natural because it's bacteria and you can just wash it off. You can also take genes for insecticides, put them into Pseudomonas organisms, and then spray that on your plants, and now you won't get insects eating the plants. Because the bacteria is there, they eat the bacteria, get the insecticide, and then die. You can use it for bioremediation. So you can manipulate prokaryotes so that they like hydrocarbons or oil. Okay? If you have an oil spill in the ocean, you release all these bacteria, they chew it up into little bits and pieces, and it gets rid of the oil. Okay? So this can be very useful in many, many different ways. In higher organisms, like eukaryotes, the most common eukaryote to be manipulated this way are going to be plants. Okay? And this is where we create transgenic plants, which often get called GMOs, genetically modified organisms. The way that we do this is we take a special bacterium called agrobacterium. This guy's kind of weird. He acts more like a virus than a bacteria. What he does is he chews through the cell wall of a plant, and then he will kind of attach himself and parasitize the plant cell. So what we do is we take out all the bad parts of that, all the parasitic stuff inside of the organism. Instead, we put in our DNA of interest. We then let him chew through the plant. He then pushes in our DNA of interest into the cell. And if we do this to a baby plant, once it grows into an adult, every single cell will have that modified DNA in it. Why is this useful? Well, we can put in things like herbicide resistance, which allow for the plant to survive in the presence of herbicides like Roundup. This becomes useful if you're a farmer, because if you have a lot of weeds taken over, you can spray the weeds, they die, but your plant of interest survives. Okay? You can put in pest protection, like insecticides, making them less tasty to insects. And you can also add nutritional value, making things more nutritional. Okay? An example where this gets done is with rice. You can add in a lot more vitamins, like vitamin A, for example. And this becomes useful when you deal with areas of the globe where rice is the only thing that they eat, okay? Because they don't have enough money to get other sources of food. And so now they can actually get a more complete diet just from eating rice. Now, it's important to note that all of this genetically modified organism stuff is 100% patentable, which means whoever creates this can create a patent for it, and then they can charge fees for people to use this. Okay? So this is a really big business for pharmaceutical companies like Monsanto, for example, because they can manipulate plants, make them better, and then sell those seeds directly to farmers. And because it's patented and licensed, that means the farmer can't take the seeds from the plants that he's grown and reuse them. Instead, he has to only buy the plant seeds from Monsanto. This is why Monsanto is $1 billion company. It's huge. Okay? So it is something that gets used a lot because it is so profitable. Now we can also do this to animals. This is a transgenic animal, and this is where we manipulate the embryo. Okay, so before it actually becomes a full-fledged organism. Now, as you can imagine, there are a lot of ethical concerns with doing this. You start doing this to animals, and you might get some weird animal that can do crazy stuff that no one wants, right? So this isn't done terribly often in the real world, but in basic science, this gets done a lot, primarily in mice and rats. Okay, so you can, add a gene or delete a gene in a mouse, and then you can actually utilize that to create like a disease model. So an example is I could go in and I could delete insulin from a mouse embryo, then grow that mouse up, and now I have a mouse that has diabetes. Now I can test different diabetes drugs and see what happens, see if they work. Okay? So this does get done, but not in a practical sense. It's more in a science sense.
So is that how the grapple came about?
Right, right. And there are, so the grapples are, it may be through cross-pollination, but there could be some genetic modification in there too. Because those are plants, so that's easy enough to do. One actual practical application of transgenic animals that actually you can go and buy right now if you really want it are glowfish. Anyone ever hear of glowfish?
Yeah.
They're fish that glow in the dark. How do they glow in the dark? We put in a protein that glows. An example of one would be GFP, right? The same protein we're going to put into our bacteria. So those little fish that you see that are yellowy green color, they have GFP in them. Okay? Those are the first organisms that are animals that are transgenic and commercially sold. Okay? Otherwise, it gets very, very difficult to do because you can screw up the organisms big time, and that could be very problematic. All right, so everything we've talked about is dealing with making protein or genetically modifying organisms. Okay. Well, what about in humans? How can we use this technique to actually help humans directly? Okay. Obviously, we're not going to make a genetically modified human, right? We're not going to manipulate embryos and create some weird, crazy baby thing. Why? Because ethically, that's bad, right? We shouldn't do that. But what we could do is use this approach to treat already full-fledged children or adults and potentially repair any genetic disorders they may already have. Okay? There are two ways that we can do this. Well, first of all, this is called gene therapy. Okay? So this is genetically modifying an organism after it's developed, and it's usually temporary. And there's two ways that we can go about doing this, ex vivo or in vivo. Ex vivo means outside of the body. In vivo means inside of the body. Ex vivo gene therapy is by far and large the most common approach that's currently being worked out. It's not commercially available yet, but a lot of these are very close to being commercially available. Okay? They're in clinical trials and they're showing great promise. These are generally considered safer because the way that it works is you pull the cells out of the patient, manipulate those cells genetically, and then put them back into the patient. Okay? So an example of where this gets used a lot is in cancer therapy. So what you can do is you can pull out immune cells from your patient, and these immune cells are designed to target the cancer, but for whatever reason they're not doing a good job, and that's why you develop the cancer. So if you pull these guys out of the body and then manipulate them genetically so that they become better cancer killers, you can then inject them back in the patient. It doesn't hurt the patient because they're the patient's cells, but now they go better or go to the site of the cancer and they fight it better than they were before. And you may ask, how do we actually get the genetic material into these patient cells? Well, surprisingly enough, it's using a virus like HIV. But what we do is instead of it being a normal HIV that can cause disease, we pull out all the bad parts of the HIV genome and replace it with whatever we're interested in. Okay? We then take that mutated version of HIV, infect the cells, and then put it into a patient. Our lab is currently working on this to actually treat HIV, which would be kind of weird, right? You're taking HIV, infecting a cell so that it goes and kills HIV in a patient. In vivo gene therapy is still pretty new. It's a lot more controversial, and that's because instead of taking the cells out and manipulating them, you're manipulating inside of the body. And as you can imagine, this is more controversial because we don't really know what's going on in the body. We're not in control of what's happening. The two approaches for doing this is to either put in naked DNA, so this is just pure vector and just sticking it right into the patient, and some of it gets into some cells just magically and then things get produced. Or to make a virus like we talked about before, but this, instead of just putting it in cells in a petri dish, you inject it directly into your patient. And so you can see why that's a little more controversial, right? You're taking a virus and you're putting it into your patient. Now this has been done before. A lot of the HIV vaccines are actually viral vectors that they inject into you. And because we delete them so they're not infectious anymore, it's really not that dangerous per se. However, there are some problems. If you use a virus that a patient has already been exposed to before, their immune system will get rid of it before it can do anything for you. Also, it could cause your immune system to react very quickly, and this could cause something called shock and be potentially deadly. Okay? So the only way that this really works is if you use viruses that humans don't get infected with easily. For the naked DNA side, it's a little safer. One example of where this is being used, at least with HIV, is you can take DNA that codes for antibodies, bind to DNA, or bind to HIV, excuse me, and neutralize it so it doesn't get you infected. And you can take that DNA and inject it into your muscle. The muscle cells then pick up the DNA, and then they start becoming antibody factories. These antibodies are released in the body, and then you're protected from getting HIV. So in theory, it sounds like a good idea. We've tested it in monkeys. It works. But it's a lot harder to go from monkeys to humans, at least as far as the FDA is concerned. Now, everything we've talked about thus far is putting genes into cells and making stuff, right? What if we want to do the opposite and we want to turn a gene off? We can do that using something called gene silencing. And this is where we either delete the gene, so cut out the DNA and get rid of it inside of a living cell, or we get rid of the RNA that is produced from that gene. So if we're getting rid of the RNA, this is called RNA silencing, and this is using something called shRNAs. And it just chews up the DNA of interest. If we want to get rid of the DNA, this is using a newer technique called CRISPR. And this is a little restriction of the nuclease, just like the one we talked about before. But you can target it to very specific regions, okay? So you can target it to just one gene inside of a cell. Once it gets in, it cuts that DNA, and then the cell tries to repair it, and it causes the gene to no longer function. Why would we do this? Well, in the basic science perspective, we do this to see what the gene does, right? If I take a cell and I get rid of a gene, and then that cell dies, it tells me that gene's important, right? Or if I get rid of a gene and it gets all fat and weird, it tells me maybe it made the cell skinny, okay? So we can infer things from what these genes do based on getting rid of them. In a clinical side of things, again, this is very new, it's not in the clinic yet, but in theory, it could be useful in getting rid of infections. So if you have someone that has HIV, you could design a CRISPR that cuts that DNA for HIV out so that it no longer is infected. Okay. Now, lastly, we can use some of these techniques to understand more about the genome itself. Okay. So in this case, we're not trying to isolate a gene and make the protein from it. Instead, we're trying to understand where the gene is in the genome, okay, and why is it there. So for these approaches, we can determine things like the locus, and the locus is the location within the chromosome of that gene. Okay, where is it found in relation to everyone else? This then allows us to determine the alleles. And the alleles are the differences between the same locuses in individuals. Okay? So my locus looks different than your locus. Okay? Each one of those are different alleles. And not only is it in different organisms or different individuals, it's also within different chromosomes in that individual. Okay? Remember, if you're diploid like ourselves, you have two sets of chromosomes, one from mom and one from dad. They code for the same genes, but the genes are slightly different. Okay, so you have two alleles of each gene in your body, one from mom, one from dad. And then we can take all of this information and come up with mapping. And mapping is just where are these genes located, okay, within the chromosome in particular. One kind of cool place where this can get utilized is in crime scene investigations. Okay? So all of our DNA is slightly different from individual to individual. Okay? For the most part, it's 99.99% the same. Okay? But there are some slight differences, and that's what makes each of us unique. So what that means is that we all have unique restriction enzyme sites. Okay? So remember, restriction enzyme sites are little pieces of DNA where an enzyme can come in and cut it. And because my DNA is different than yours, maybe in my DNA it cuts right here, and in yours it cuts over here. Just because our DNA is slightly different. So what that means is if I take all of our DNA and I cut it with the exact same enzymes, and then run it through a gel, all of us will have different bands that get produced. Because they cut at different places and they became different sized fragments. This is what we call a DNA barcode. This is unique for each individual. Where does this get used? Crime scenes. Okay? So in this example down here, we have a victim. Okay? And here's the victim's DNA, just to make sure that we're not looking at the victim's DNA, we're looking at the suspect's. And at the crime scene, we found evidence that had DNA on it, and we were able to cut that DNA, and this is the pattern that developed. So you can see these bands that formed. Okay? We now have two suspects that we think may have committed the crime. We take their DNA, we cut it with the same enzymes, we run it on a gel, and you get banding patterns that look like this. Out of those two suspects, who do you think committed the crime? Suspect 1, right? Because their bands are exactly the same. And again, that only can happen if it's the same DNA. Now you can't prove that it's them because maybe they were just in the apartment cleaning the carpets the day before or something like that, but at least it points you in the right direction. Okay? And don't worry about that. Alright, that's it for chapter 10. Any questions? The biggest thing that I want you guys to understand is PCR, and we'll go over it again before the next